A 34-year-old patient presents with fever and cough. A nasopharyngeal swab is sent for SARS-CoV-2 RT-PCR. The result comes back inconclusive. Not negative, not positive. Before you blame the virus, the pathogen, or the machine, ask yourself, what actually happened to the RNA before it ever reached the PCR tube? Every molecular diagnostic test, every RNA sequencing experiment, every mRNA vaccine batch release, all of it begins with one unglamorous, invisible step. Get it wrong, and everything downstream fails silently without ever telling you why. RNA extraction is the process of isolating ribonucleic acid from cells, tissue, blood, or a viral sample, separating it away from proteins, DNA, lipids, and salts, while keeping the RNA itself chemically intact. It sounds simple. Biochemically, it's a race against time and enzymes. Not all RNA is the same, and not every extraction targets the same molecule. Messenger RNA carries the protein coding blueprint. Ribosomal RNA makes up the bulk of total RNA and builds the ribosome itself. Transfer RNA ferries amino acids during translation. And viral RNA, the genome of pathogens like SARS-CoV-2 or influenza, is exactly what diagnostic RT-PCR is built to detect. During the pandemic, a landmark study by Kusurka and colleagues, published in Annals of Internal Medicine in 2020, showed RT-PCR false negative rates varying dramatically by day since exposure, sometimes exceeding 20 to 30% even at optimal testing windows. Sample collection and extraction quality are consistently cited as major contributors to that false negative gap. This one technique feeds an enormous range of downstream methods: reverse transcription PCR for diagnosing infections, RNA sequencing for gene expression profiling, northern blotting for transcript-specific detection, microarrays, and quality control testing during mRNA vaccine manufacturing. Every one of these techniques inherits whatever quality or damage happened during extraction. RNA extraction shares its logic with DNA extraction, lysis, separation, purification, but the resemblance ends there. RNA is single-stranded, chemically fragile, and under constant enzymatic attack from ribonucleases that are essentially everywhere. On your skin, in your breath, on unwashed glassware. DNA extraction rarely has to fight this hard just to survive the protocol. High-yield exam pearl. Examiners love to ask why RNA extraction is considered the rate-limiting, error-prone step of molecular diagnostics. The answer isn't the PCR chemistry, it's that RNA quality is invisible until the assay fails, and by then, the sample may be unrecoverable. So before we ever touch a PCR machine, we need to master three things: why RNA is so fragile, how the extraction chemistry actually works, and how to prove quantitatively that what we extracted is worth using. Ribonucleases, or RNases, are enzymes that cleave the phosphodiester backbone of RNA. They're not rare lab reagents, they're everywhere. On your fingertips, in your saliva and breath, on dust particles, even trace amounts, invisible and odorless, can shred an RNA sample in minutes. Mechanistically, most RNases act as endonucleases, cleaving internal phosphodiester bonds via a 2'3" phosphate intermediate, a reaction made possible by RNa's free 2-hydroxyl group, which DNA simply doesn't have. One active RNase molecule can cleave thousands of RNA strands before you've even finished pipetting your sample. Here's a fact that surprises most students. RNase A, one of the most common contaminating RNases, is famous for being extraordinarily heat-stable. Its disulfide bonds allow it to refold and regain activity, even after boiling, a property first characterized in Christian Amphenson's classic protein folding experiments. Autoclaving alone often isn't enough to destroy it. This is why RNA work demands almost ritualistic discipline. RNase-free, DEPC-treated water. Dedicated RNase-free plasticware and pipette tips. Fresh gloves changed frequently. Decontaminating benches and pipettes with commercial RNase eliminating sprays, and keeping every sample on ice from the moment of collection to the moment of freezing. The most common source of RNase contamination isn't a mysterious reagent. It's the researcher. Bare-handed pipetting, reusing DNA-grade plasticware, talking over an open tube, and letting a sample sit at room temperature are the leading causes of degraded RNA in student and research labs alike. In a molecular diagnostics lab, a proper aseptic RNA workflow starts before the sample ever arrives. Designate an RNase-free zone, use a fresh set of gloves per sample, wipe the workspace with a decontaminant, and never let an open tube sit unattended. Exam Pearl and a genuinely useful mnemonic. Think fresh for RNA extraction discipline. Fresh reagents and tips, RNase free zone, every tube on ice, single glove change per sample, handle fast, minimize freeze-thaw cycles. Respect the enzyme. RNases don't need a lab accident to ruin your sample. An unwashed hand or 1 warm minute on the bench is more than enough. Regardless of which specific kit or reagent you use, every RNA extraction protocol follows the same 4 core stages. Lysis of the cell or viral envelope, rapid inactivation of RNases, separation of RNA away from protein, DNA, and lipids, and finally purification and elution of the RNA into a clean buffer. The workhorse molecule behind lysis and RNase inactivation is guanidinium thiocyanate, a powerful chromotropic salt. It disrupts hydrogen bonding and denatures proteins in a single step, ripping apart cell membranes while simultaneously unfolding and inactivating any RNase enzymes it encounters, before they get the chance to attack your RNA. Why is RNA less stable than DNA in the first place? The answer is a single hydroxyl group. RNA carries a free 2' OH on its ribose sugar. DNA, lacking that oxygen at the 2 position, does not. That extra 2' OH makes RNA both more chemically reactive to hydrolysis and the exact structural feature RNases exploit to cleave it. Once lysis is complete, the mixture must be separated into distinct phases. In organic extraction, adding chloroform and centrifuging creates 3 layers. Which layer contains your RNA, and which contains the DNA and protein you don't want? Getting this decision wrong at the bench is one of the most common practical mistakes students make. Before lysis, RNA sits tightly bound within an intact cell, wrapped in ribonucleoprotein complexes, protected by an intact membrane, and shielded from the outside world. After lysis, all of that protection vanishes in seconds. The membrane is destroyed, proteins are denatured, and the RNA is suddenly free-floating and vulnerable in solution. Not all RNA molecules are the same size, and this matters for extraction chemistry. MicroRNAs are barely 20 to 24 nucleotides long. Messenger RNAs range from a few hundred to several thousand nucleotides. Ribosomal RNA subunits like 18S and 28S can reach nearly 5,000 nucleotides. Kits optimized for total RNA can lose the smallest species entirely if not designed to retain them. After phase separation, the aqueous RNA-containing layer is carefully transferred to a fresh tube. Then RNA is precipitated using isopropanol, pelleted by centrifugation, washed with ethanol to remove residual salts, air-dried briefly, and finally resuspended in RNase-free water or buffer. This structural comparison is worth memorizing cold. RNA is single-stranded with a 2' OH and uses uracil instead of thymine. DNA is double-stranded, lacks the 2' OH and uses thymine. Every downstream decision from which chromotropic agent to use to how gently you pipette traces back to these 3 differences. Exam Pearl. If a question asks why RNA is more prone to alkaline hydrolysis than DNA, the answer is always the free 2' hydroxyl group. It attacks the adjacent phosphodiester bond intramolecularly, something DNA's chemistry simply cannot do. For stages, one guiding principle: move fast, keep it cold, and never let the RNA sit exposed longer than it has to. In 1987, Pyotr Chomczynski and Nicoletta Sacchi published a single-step method in analytical biochemistry that would become the gold standard foundation for RNA extraction for decades. Their acid guanidinium thiocyanate-phenol-chloroform method was later commercialized under names like thiarizol, and it remains a benchmark technique taught in labs worldwide. The protocol itself follows a precise sequence. Add the guanidinium phenol reagent to lyse and denature. Add chloroform and shake vigorously. Centrifuge to separate 3 phases. Carefully transfer only the clear aqueous top layer. Precipitate the RNA with isopropanol. Wash the pellet with ethanol, and finally resuspend in RNase-free water. Phenol and chloroform work as a team. Phenol denatures and dissolves proteins, pulling them out of the aqueous phase. Chloroform sharpens the separation between phases and helps remove residual phenol from the final aqueous layer, which is critical. Leftover phenol will contaminate your downstream reactions and inhibit your PCR. Before centrifugation, the tube is a single cloudy, homogeneous mixture: cells, proteins, DNA, and RNA all suspended together. After centrifugation, 3 sharply distinct layers appear. A colorless aqueous top layer holding your RNA, a white interphase holding DNA and denatured protein, and a pink organic bottom layer holding lipids, protein, and phenol-chloroform. Each of the three phases has a very distinct appearance and a very distinct danger if you contaminate your sample by pipetting too deep. The aqueous phase is colorless and holds your RNA. The interphase is a thin white layer holding genomic DNA and denatured protein, and the organic phase is pink from the phenol-chloroform, holding lipids and remaining protein. Weigh the trade-offs honestly. The phenol-chloroform method delivers excellent yield and works on almost any sample type, from tissue to viral culture. But it uses toxic, hazardous reagents requiring careful disposal. It's labor-intensive and time-consuming, and manual handling introduces more opportunity for RNA degradation than an automated kit. Quick pause. If this breakdown is helping you, take a second to hit like and subscribe. It genuinely takes a lot of effort to make videos like this, and your support keeps them coming. Exam Pearl. Guanidinium thiocyanate is the reagent name examiners most often test. It's the chromotropic salt responsible for simultaneous cell lysis and RNase denaturation, the single molecule that makes the entire single-step method possible. The phenol-chloroform method is a rite of passage in molecular biology training, powerful, reliable, but demanding total respect for both the chemistry and the chemicals involved. Compare a manual phenol-chloroform extraction, hours of pipetting, toxic waste, careful phase transfers, against a modern silica-column or magnetic bead kit, which can deliver purified RNA in as little as 15 to 20 minutes, with far less hands-on risk of contamination. Silica spin column extraction follows its own workflow. Lyse the sample in a chromotropic buffer, load the lysate onto a silica membrane, wash away contaminants with ethanol-based buffers, dry spin to remove residual ethanol, and elute the purified RNA in a small volume of RNase-free water. The chemistry behind silica binding is elegant. In the presence of a high-salt, chromotropic buffer combined with ethanol, RNA's negatively charged phosphate backbone forms hydrogen bonds directly with the silica surface. Lower salt ethanol washes then strip away protein and salts while the RNA stays bound, until a low salt-elution buffer releases it back into solution. Every column-based protocol moves through the same recognizable stages. Load, wash once, wash twice, dry spin to remove ethanol traces, and elute. Skipping the dry spin step is one of the most common student errors. Residual ethanol carried into your eluate will directly inhibit downstream RT-PCR. Magnetic bead-based extraction takes the same silica-binding chemistry and attaches it to microscopic paramagnetic particles suspended in solution. A magnet pulls the RNA-bound beads to one side of the tube, letting you wash away everything else by simply changing liquids, no centrifuge required. This is exactly what makes it fully automatable. The throughput difference is dramatic. A manual phenol-chloroform workflow typically processes a handful of samples per hour. A silica spin column kit can push that closer to 2 to 3 dozen samples per hour. Fully automated magnetic bead platforms, running on liquid handling robots, can process 96 or more samples in a single run. 3 broad classes cover almost every extraction method you'll encounter. Traditional organic extraction, best for maximum yield and flexibility across sample types. Silica column kits, the standard for small research or clinical batches, and magnetic bead-based systems, purpose-built for high-throughput diagnostic labs and public health surveillance. So how do you actually choose? A research lab running a handful of gene expression samples a week can comfortably use a column kit. A hospital diagnostic lab running hundreds of RT-PCR tests daily during an outbreak needs magnetic bead automation. And a forensic lab working with a trace, degraded sample may still reach for phenol-chloroform's raw sensitivity. No single method is universally best. The right choice always depends on your sample, your scale, and your turnaround time requirements. Extraction isn't finished until you've proven the RNA is both pure and intact. The first and most classic metric is the A260 or 280 absorbance ratio. Pure RNA should sit around 1.8 to 2.1. A ratio below 1.8 signals residual protein contamination, one of the most frequently tested numbers in a molecular biology practical. The concentration itself is calculated using a standard formula rooted in the Beer-Lambert law. RNA concentration in micrograms per milliliter equals the A260 reading multiplied by 40, the extinction coefficient for single-stranded RNA, multiplied by the dilution factor. A 260 or 280 tells you about protein contamination, but it's blind to salts, phenol, and carbohydrates. That's where the A260 or 230 ratio comes in, also expected around 2.0 to 2.2. A low A260 or 230 is the classic giveaway for leftover guanidinium salts or residual phenol, both of which will inhibit your downstream PCR. In practice, labs often quantify the same sample two different ways, spectrophotometrically on a nanodrop and fluorometrically on a qubit, and compare the results. Fluorometric methods use RNA-specific dyes and are far less fooled by contaminants, so a large disagreement between the two readings is itself a red flag worth investigating. Concentration and purity say nothing about whether the RNA is fragmented. For that, run a denaturing agarose gel and look for two sharp ribosomal bands, 28S and 18S, at roughly a 2:1 intensity ratio. A smeared trace instead of clean peaks is the classic signature of degraded, unusable RNA. Because visually judging gel bands is subjective, the field moved toward the RNA Integrity Number, or RIN, a Bayesian algorithm-scored value from 1 to 10 introduced by Schroeder and colleagues in 2006, using the full microcapillary electropherogram trace rather than just two bands. Most downstream applications like RNA-seq require a RIN of 7 or higher. For a quick viva reference, memorize this cluster of benchmark numbers together: A260 or 280 around 1.8 to 2.1, A260 or 230 around 2.0 to 2.2, a 28S, 18S ratio around 2.0, and a RIN of 7 or above. Any one of these falling short should trigger a re-extraction before you invest time in downstream work. Exam Pearl mnemonic for the QC panel: ratio, RNA conc via A260 times 40, A260 or 280 for protein, 2:1 28S, 18S ratio, integrity number, RIN, greater than or equal to 7, other contaminants via A260 or 230. You haven't finished an RNA extraction until you've proven, with numbers, that what's in the tube is both pure and intact. Zoom out to the pandemic scale pipeline: sample collection, RNA extraction, RT-PCR amplification, and result reporting. During surges, extraction was consistently the throughput bottleneck, the step limiting how many results a lab could report per day, driving the mass adoption of automated magnetic bead platforms worldwide. Back to our opening patient. The repeat sample was re-extracted using a fresh silica-column kit with a verified A260 or 280 of 2.0 and a RIN of 8. And this time, the RT-PCR returned a clear, confident positive result. The virus never changed. The RNA quality did. Beyond diagnosing infections, high-quality RNA extraction underpins liquid biopsy, detecting circulating tumor RNA in a cancer patient's blood, newborn metabolic and genetic screening panels, quality control batch release testing for mRNA vaccines, and forensic identification of tissue or body fluid type from degraded crime scene samples. Not every application needs the same grade of RNA. Diagnostic RT-PCR can tolerate mild fragmentation as long as the short target sequence remains intact. RNA sequencing demands high integrity, RIN 7 or above, across the entire transcript. Forensic RNA work often has to succeed with severely degraded material, using specially designed short amplicon assays. Across the published methodological literature, one finding repeats consistently: samples extracted with poor A260 or 230 ratios or low RIN scores show measurably reduced RT-PCR sensitivity and higher CT value variability, directly reinforcing why QC isn't optional, it's diagnostic infrastructure. Reference grade viral RNA, used to validate new RT-PCR assays and calibrate diagnostic kits, is typically extracted directly from a controlled viral culture, under strict biosafety containment, giving labs a known quantity, high purity standard to test every new diagnostic method against. Final high-yield exam cluster: pure RNA reads A260 or 280 around 2.0 versus pure DNA at around 1.8. A favorite, spot the difference question. RNase survives boiling. Thiorizol style extraction is Guanidinium thiocyanate-phenol-chloroform, first published by Chomczynski and Sacchi in 1987. Practice question. A student measures an A260 or 280 ratio of 1.65 on their extracted RNA sample. What does this most likely indicate? Quick recap before you go. What enzyme is the biggest threat to RNA samples? RNase. What salt lyses cells and denatures RNase in one step? Guanidinium thiocyanate. What A260 or 280 ratio signals pure RNA? Around 2.0. What RIN score is the cutoff for RNA-seq? 7 or above. Standing back, RNA extraction is a genuine trade-off. Hard-won reliability requires strict discipline and careful technique, but skip the discipline, and every downstream result, clinical or research, becomes questionable. RNA extraction may be invisible in the final result, but it determines whether that result can be trusted at all. 3 things worth carrying into your next practical. RNases are everywhere, so work fast and cold. Match your extraction method to your sample and scale. And never trust a yield until A260 or 280, A260 or 230, and RIN all check out. Here are the key references and sources for this video. Thanks so much for watching. If this helped, like the video and subscribe for more clear, simple explainers, and I'll see you in the next one.